This lecture will cover DNA metabolism, and the title slide is kind of a good overview of what we're going to be talking about today, DNA replication. And it doesn't show everything. It's missing the topoisomerases, but it gives a good view of everything else that goes on in replication. You have a helicase, which is unwinding the DNA, and then you have DNA polymerase on each strand. What's called the leading strand just continues to get replicated in the direction that you're unwinding the DNA, and the lagging strand, as you can see, is going the opposite direction, right? But let's say you start here and move in this direction. As this unwinds further, you can see that you're going to have other polymerases fill in the new unwound DNA, and so there's a way that the cell handles this that we'll talk about during the lecture. Okay. So the last three lectures of this semester, we're going to be covering chapters 25, 26, and 27, and this is really what I consider to be where we cover the central dogma of biochemistry, and that's DNA information being converted to DNA, being converted to RNA, and being converted into protein. And the chapter titles are shown here for 25, 26, and 27. And the terms they use, I don't really like them. DNA metabolism, it doesn't necessarily tell you what we're really studying. I think it really should be titled replication, personally, but it's not. RNA metabolism in chapter 26, what we're really covering there is transcription. In the protein metabolism in chapter 27, what we're really covering there is translation. Okay. And all three of these lectures have quite a few slides in them. I'm going to move quickly through them. And so really, I included a bunch of slides so that you can have in a handout. They're available on Canvas, and you can kind of go through them to study. And I think it's good to have all the information there, but I'm not necessarily going to be spending a whole lot of time on each slide in these lectures. All right, so let's talk about some conventions. So for prokaryotic genes, there's a three-letter abbreviations, and they tell the function of that gene. Okay, they're in lowercase and italic, and then they're followed by a capital letter. The protein name is the same as the gene, but it's not italicized, and the first letter is also capitalized. So for example, a prokaryotic gene will be written as pro-A, right? So the three letters at the start are lowercase, and the last one is a capital letter, and they're italicized. The protein of that same gene would be written pro-A but not italicized, and the first letter would be capitalized. So that's just something to look for when if you're dealing with genes and protein products of genes. For instance, if you're reading a paper, let's say. And those are for prokaryotic genes. Eukaryotic genes are a little bit more complicated. The abbreviations have a different number of letters. They can have 4 is the most common, but they can have a different number of letters. Usually they tell the function of that gene, but not always. Okay, it kind of depends on when the gene was discovered and what circumstances it was discovered. They use uppercase letters and italics for the gene, so that's important to note that prokaryotic genes are lowercase, eukaryotic genes are uppercase, but both are italicized. So if you're reading a paper and you see something italicized, that's talking specifically about the gene. Okay, if it's not italicized, it's talking about a protein. Okay. Sometimes they're followed by numbers, right, depending on what gene you're looking at. Okay, so for example, pol A1 would be the gene for a polymerase, and pol alpha is the protein. So the gene and the protein don't always, abbreviations don't always match up. All right. Well, historically, if we look back in history, it was in the middle of last century when it was really discovered that DNA was the genetic material, and that there's a famous experiment where you inject a mouse with a non-virulent rough strain of a virus, and that mouse would live, or I should say bacteria, and then you inject a smooth strain that is the virulent into the mouse, and the mouse would die. If you heat killed that smooth strain and inject it, the mouse would still live. And if you mixed the rough strain and a heat-killed smooth strain and mixed that and injected it into the mouse would die. So there was something from that heat-killed smooth strain that the rough strain was picking up and allowing it to gain virulence. And that was the DNA. And then through some other experiments using radioactivity, they really, it was narrowed down to DNA that was passing on this genetic information. And not too long after that, 53, the structure of DNA was resolved by Watson and Crick. Okay, and so once the structure was found, there was a lot of other important biochemical questions to tackle. Okay, and so we're going to go through some of these questions. And specifically in Chapter 25, what we're going to cover is the replication of DNA, which is in Section 1, errors that arise during DNA replication, how they are repaired, and that would be in Section 2, and then how DNA gets rearranged in a chromosome, and that'd be in Section 3. And I would say by far the most important section in this class is the first section, understanding DNA replication. I think that's definitely the most important. Right. So back to Watson and Crick, they discovered the alpha, I shouldn't say alpha helix, the double helix of DNA, and through crystallography. And once this structure was determined, the next sort of questions that arose was, okay, how does the cell use this to store and transfer information to the new generation of cells? Right. So we know that the DNA has to be duplicated, replicated before cells can divide. And is that replication semi-conservative or conservative? Does the replication proceed in one direction, or does it proceed in both directions? Okay, are both strands copied at the same time, or one strand copied and then the next strand? Are the parent DNA strands completely unwound before each is replicated, or does the unwinding and replication occur at the same time? Does replication begin at random places or at unique points? Okay, those were the real questions that needed to be answered. Okay, so the first one, is it conservative or semi-conservative? Okay, what conservative would mean is that the parent strands would be replicated, and when they're replicated, the daughter strands are bound to each other. So when you have, let's just say this is a chromosome, when you replicated that chromosome, the parent strands are back together, and the newly synthesized daughter DNA is both together. Semi-conservative would be the opposite, where each parent strand has a new daughter strand that has been synthesized. All right, and this experiment really nailed down the answer to this question. So what they would do is they used a growth medium containing an N15 isotope. In DNA, those bases have nitrogen in them, so that the DNA would become fully labeled with that N15. And then they switched it to an N14 medium and allowed the cells to divide once. When they did this, I don't want to go through all the details here, but basically what they found is that you'd get after the first generation of daughter molecules, you'd get this 50-50 mixture of N15 and N14. And so that meant it was a semi-conservative, the replication occurred in a semi-conservative manner. Okay, and that is for each parent strand, a new daughter strand would be synthesized onto it. Okay. The next question is, where is replication taking place? Does the replication proceed in one direction or both? Well, it is bidirectional. So if you look at this under a very powerful microscope, you can see that the DNA, and this is DNA being replicated, you can see that there's what's called a replication bubble. So the DNA strands are unwinding here. So this would be the parent strands together. They're unwinding, and then they're being synthesized on each of these parent strands. The next question, are both strands copied at the same time? And yes, the answer to that is yes. And that was found out by using radioactive isotopes labeled into the newly incorporated DNA. Okay. And it turns out the replication is actually occurring at 4 places. So you have it going this direction and that direction, and it's occurring on both strands. So that's 4 separate places. Here's the figure from the book showing a very similar thing where you have what's known as the replication forks in the replication bubble, and exactly what's happening and how it's happening with the blue parent DNA and the newly synthesized orange daughter DNA. So that's a very, very, I guess, simplified overview of how this is occurring. Okay, so replication is semi-conservative. It proceeds in both directions. Both sides, both strands are copied at the same time. Okay. Are the parent DNA strands completely unwound before each is replicated? Does the replication begin at random places at unique points? Well, to answer this, there was research found that there's actually origins of replication. And when you look at like a bacterial plasmid or chromosome, you see that ORI block there on some of those. That stands for origin, and that's an origin of replication. So the replication will start there, and it proceeds in both directions. Eukaryotes have several origins of replication, so their genomes are a little bit more complicated, bigger, and they have several origins. Not really wanting to get into a lot of detail here, but this shows origin of replication in binding sites for the DNA A protein, which initiates unwinding and replication. And so there are multiple sequences going in both directions there for those DNA A proteins. Okay, and the consensus sequence is shown there. Again, probably not a consensus sequence that you need to remember. Okay, so back to our questions that we're asking. are parent DNA strands completely unwound before each is replicated? Okay. We've answered question 5. Does replication begin at random places? No, it's unique points. Right. And so the question on whether DNA is unwound completely before it's replicated, and you might kind of already know this, or just logically it makes sense that it's not completely unwound before it's replicated. All right, that the process of unwinding all the DNA, then replicating it would take longer. So as it's unwound, it's being replicated, and that's where we can see this bubble. Okay, so it's being unwound in both directions here, and as that's being unwound, it is being replicated. And to do this, there's a number of different things that are needed, right? More than just a single DNA polymerase. Okay, you need to do the separation of strands. You have to maintain them as single strands, right? So once you unwind this, what they would want to do, these two strands, they'd really want to bind back together because they're complementary, right? So you have to keep them maintained as single strands. Okay, you need primers before you can synthesize DNA. You need a small primer that matches up to your DNA that you're replicating. You want to your DNA polymerase needs that primer to tell it where to start. You need to remove and replace the primer as needed, and then there'll be nicks, or as we'll see on the lagging strand, there'll be places that the sequence, the newly synthesized sequence, isn't completely attached, that need to be, and those little nicks need to be sealed. And this occurs in 3 stages: initiation, elongation, and termination. Right, so initiation is where you have the origin of replication, where you have these DNA proteins binding. So DNA A proteins bind to these, and this is specifically in a prokaryotic system here. You bind to these 5 9-base pair repeats, right? When DNA A binds, it will denature those AT-rich sequence, and that's the DNA unwinding element, which is shown in yellow. Okay. DNA B, which is also known as helicase, that separates the duplex DNA beginning in the DUE region. SSB is a protein that's a single-stranded binding protein. That's what SSB stands for. And that just helps bind to the single-stranded DNA and prevent it from coming back into double-strand. Okay, and then you have a DNA gyrase, which is, if you remember back from last lecture, DNA topoisomerase 2, which helps as the DNA is being unwound, it can bind up, and that helps cut it and alleviate the pressure from unwinding. So if we look at this in a picture, right, DNA A first binds to that origin of replication. Okay, when that happens, what occurs is you get a lot of these DNA A proteins binding in that region, and it helps it get some sort of a structure to it there. And when it does that DUE region can unwind. Right? And when that unwinds, now DNA B, the helicase, and DNA C can help it. We didn't really talk about DNA C, but the DNA B will go into that and be on both ends of that bubble that this is forming. Okay, and remember, DNA B is known as helicase. It's the protein that is responsible for unwinding the DNA. Okay. So which direction does elongation occur? Well, as you're unwinding this DNA, you can think of one of the strands will be going 5' to 3'. And if you remember back from our discussion on nucleic acids, and we talked about PCR, that's the direction that DNA is going to be synthesized. Well, if one strand's 5' to 3', right, that's good. That means you can just synthesize the DNA on that strand as you're unwinding it. The other strand's going to be the opposite. Okay, it's going to be backwards. So there are going to be differences in how each strand is synthesized and replicated. Right. The synthesis in DNA always occurs with the addition of nucleotides to the 3' end. Okay, that 3' hydroxyl is where the new nucleotide will be added. Okay. The leading strand is made continuously, so the strand that's in that direction, with respect to how you're unraveling the DNA, that's called the leading strand. Okay, and it's made continuously. And that's as the replication fork advances, you're continuously adding to that leading strand. The lagging strand is the opposite. Okay, and if you think about it, if you unwind a little bit and you start polymerizing that backward strand, you can do it from there. That's fine. But as you unwind further, you're going to have bare single-stranded DNA again. So you're going to have to continuously keep starting that polymerase. And those make, what would appear to be fragments. And those are called Okazaki fragments. Okay. After, they're made, they have to be joined. Right. So as we're doing this, the chemistry involved in DNA elongation, the parental strand is providing the template. What else you really need are nucleotide triphosphates, because they're serving as the new substrates to your polymerase enzyme. Okay. The hydroxyl at the 3' end, that's the good nucleophile, and that can allow the chain to grow using the alpha phosphorus of the new nucleotide triphosphate that comes in. And then the two phosphate groups, the pyrophosphate, that's what if you have a nucleotide triphosphate, when you have a nucleophilic attack by the 3' hydroxyl to that, you're going to be losing two phosphate groups, and that's referred to as pyrophosphate. That's a very good leaving group. Right, so cartoon depiction of how this is occurring. This is the replication fork. Parental DNA is shown in blue. Our newly synthesized DNA is shown here in that orangish red color. So we're going 5' to 3' in synthesis of that leading strand. Okay, in the parental strand, you're reading it in the 3' to 5' direction. Okay, and this replication fork is moving in this direction, so this would be continuously unwinding. Okay, and as you do that, this can just continue to be polymerized, the leading strand. The lagging strand, right, it's going in the opposite direction. So if you're synthesizing 5' to 3', let's say you've unwound to this point, you can synthesize 5' to 3' in this direction, but as soon as this gets unwound further, you're going to have to start another synthesis. Okay, so that's why you have these Okazaki fragments there on the and that's termed again the lagging strand. You're going in the opposite direction as the fork. Okay. Right, so this is same figure, just slightly different colors, and it's showing that electron micrograph that we saw earlier of this DNA. Right, these are the replication forks. So this here is kind of depicting what's happening right here in this picture. All right, so what's essential to this reaction? Well, you need a template. Your template is the blue parental DNA. You need deoxynucleotide triphosphates, right? That's the substrate that your polymerase uses. You need a primer. Usually it's an RNA primer, so those are nucleotide triphosphates for that primer. And then the replisome, the proteins involved in the replication. You need a DNA polymerase. You need topoisomerases to help alleviate the tension of unwinding. You need a ligase to stick the Okazaki fragments back together at the end. You need a helicase to unwind the DNA. And then a primase just makes the primers. And specifically, you need many more primers for the lagging strand. So that's where that becomes really important. The reaction is actually pretty simple. If this is our growing DNA strand here, there's going to be a free 3' hydroxyl group. The new nucleotide, and this is all catalyzed in the polymerase, the new nucleotide that matches up with our template strand will come in, and it's in a triphosphate form. This free 3' hydroxyl will undergo nucleophilic attack on that alpha phosphate, the first phosphate group, right? And that nucleophilic attack will make this the beta and gamma phosphates leave together. Okay, so that's the pyrophosphate. Right, so in terms of how mechanisms go, this is a pretty simple mechanism. Right, and the thermodynamics of this, if you think about it, it's almost the same as if you have an ATP, for example, and you're cleaving that. That's a very exergonic reaction, and the same for this, because you're actually losing this pyrophosphate group. So it's a very favorable reaction. Okay, so here's a depiction of how this polymerase replisome is working. Okay, this is polymerase 3 from E. coli. There are actually 17 subunits to this, so it's very complicated. You don't have to know every piece of this, but knowing just the basics of how it's working is important. So remember, DNA B is the helicase. This is what's unwinding the DNA. Okay, as it does that, the leading strand can just fit through one of the core pieces of the polymerase. Okay, and there are these clamp proteins too that hold it in place. Okay, and so this core of the polymerase is where you're getting the replication occurring. Okay. The lagging strand's a little bit different. The lagging strand doesn't necessarily go directly through that core in a straight line. You get a little bit of overhang here, okay, to go through. And this lagging strand, remember one of the things that's required to start polymerization is a primer. So you have to put a primer down, and those are shown here in green, okay, RNA primers. And then you can get synthesis, okay? But again, as this unwinds more, you're going to have to keep adding primers and keep synthesizing that DNA. And that's where the primase enzyme comes in. Okay, primase binds to the DNA B right here, the helicase. It adds a new primer, RNA primer, and then that can then be used for synthesis for the lagging strand. Right, and those clamps that they're referred to as the beta clamps. Right, as you're going through on the lagging strand, those get discarded from time to time, and new ones with the new primer for the new Okazaki fragment that's coming up will get connected to the polymerase. This is a good link if you want to see this in action. I will leave this in the description box. DNA synthesis is catalyzed by the DNA polymerase. Magnesium coordinates the alpha phosphate, and it also stabilizes the negatively charged phosphate group and transition state. So many times in the cell, when you have phosphate groups and triphosphate groups, you're going to have magnesium 2+ cations around because it helps shield those charges. This is just a picture, a little bit more detail of something that we've kind of looked at, the mechanism of this, how the polymerase works. But if we have our template strand and our complementary base pairing, right, that free 3' hydroxyl is the one that's going to allow nucleophilic attack there to the incoming nucleotide triphosphate, right? But this, in the polymerase, they're magnesium ions that are helping stabilize that. And aspartic acids as well that help coordinate those magnesium ions, and those aspartic acid residues are very conserved. So this whole process of the polymerase replicating DNA has very high fidelity. Okay, and so what fidelity is, it's just a measure of how accurate something is in replicating. You might have heard that term hi-fi, which stands for high fidelity, and so that's usually associated with like speaker equipment, and that's how clear and accurate that speaker equipment or stereo can replicate the sound of a song. Right? In E. coli, the mistake frequency is something like 1 mistake in every 10 to the 9th or 10 to the 10th nucleotides added. So that's, like 1 in a billion or 10 billion, right? That's a very, very low number, right? So if you have a chromosome, a typical chromosome in E. coli, 4.6 times 10 to the 6 base pairs, that's, like 5 million base pairs. So you can think of that with the error frequency. You're going to make an error once every 1,000 to 10,000 times that cell replicates. So that's a very, very small number when you think about copying something that is 5 million letters. You're only going to make a mistake once every 1,000 times you copy that. Why is that error rate so low? Well, first you have the H bonding, right? The complementary base pairs, how they hydrogen bond together is pretty specific. Okay, so that drives some of this. Okay. You have the sterics of the active site of the DNA polymerase, right? So only certain nucleotide triphosphates can fit one, let's say one at a time, and then you also have that, matching up to the complementary strand. Do the H bonds fit? The DNA polymerase is actually putting in incorrect nucleotides once every 10 to the 4th or 10 to the 5th times. So its error rate is quite a bit higher than the one that's observed. So there has to be some sort of proofreading mechanism that's going on. And that turns out to be what's known as an exonuclease. Right? And it also has this built-in repair mechanism, even if that for that exonuclease. Right, so if you think back, we talked about endonucleases before when we talked about cloning. And endonucleases, those are what we would term restriction enzymes. And so if you remember back, you have a specific sequence. The endonuclease can cleave those phosphodiester bonds between certain base pairs in a sequence. So endo stands for within, and nuclease just means you're cleaving phosphodiester bonds between adjacent nucleotides. Okay, what an exonuclease is you cut nucleotides one at a time from the end of a polynucleotide chain. So for instance, your polymerase put in a mistake, a base pair that doesn't match. If you have a proofreading on it and it determines that was a mistake, the exonuclease can cut that one base pair off. Right, so just going through graphically some of these conditions to keep high fidelity. If you think of the polymerase active site as this blue rectangle here, right? If you have the correct matches, like an A to T or a C to G, they fit entirely within that active site. But once you start getting some of these mismatched base pairs where they can still hydrogen bond together, they just don't hydrogen bond in the Watson-Crick method, you see that they start spilling out of that blue rectangle. So they kind of move outside of that active site. Okay, so that active site helps stabilize the correct base pairing and prevent some of these incorrect base pairing. Right. The errors that are made during synthesis are going to be corrected by the 3 to 5 prime exonuclease activity. So if you look at this and this is not a very specific picture, but this is our polymerase, right? The polymerase core would be on this side. Our proofreading site would be here on the outside. And the picture in the book kind of depicts this in like 2 different colors. So this blue color is the DNA polymerase active site, and the orangey color is that 3' to 5' proofreading exonuclease site. Right, so let's say a C comes here and binds to A in that polymerase site. Well, that's not really the base pair that should be binding, but you can get this tautomerization of C that does pair with A and can be incorporated into that strand. And that's shown here, right? The tautomerization can make C look like this, which looks sort of like thiamine, right? So it can then H bond with A. Okay. Before the polymerase moves on, this cytosine then undergoes that shift back to the regular form of C. The nucleotides then are now mispaired. Okay? And the enzyme kind of senses it because it moves it back into that exonuclease site, and that mispaired base pair can then be cleaved. Right, so that bonding is cleaved, and then it removes that base on the 3' end, and then this can then be shifted back into the polymerase site where the correct nucleotide can then be added. A protein that's needed for the lagging strand. Okay, so the leading strand's pretty self-explanatory. This process just kind of occurs continuously. But for the lagging strand, as we've seen those Okazaki fragments, there'll be little nicks between them where they're not connected. And so those need to be put together with a DNA ligase. DNA polymerase 1 does perform that nick translation, which replaces the RNA primers with DNA. And then you need a DNA ligase to connect those fragments together. And remember, the primers are RNA, so you don't want these little sections of RNA within your DNA, and that's where this DNA polymerase I will replace those primers with DNA. So if we're looking at our lagging strand here, we'll have two different fragments, and the spot between them would be referred to as a nick, where they're not connected together. That green, again, is RNA. That's the primer. A DNA polymerase I, it will go through there and turn that, replace that RNA with DNA. Okay, you still have that nick, and that's where our DNA ligase comes in. It uses ATP, and it just connects those two. Right, and DNA polymerase I there has that 5' to 3' exonuclease activity. That's what's actually being used to remove that RNA primer. The DNA ligase reaction, probably not super important that you know this, but this is the reaction. First, you have your enzyme that you attach to your AMP residue here, which is you get from an ATP. Okay, that gets stuck onto your ligase, and then from there you switch that phosphate group from your ATP into that ligation. Okay, and it's a little bit complicated, but you don't really need to know this mechanism. All you really need to know is that your ligase is using ATP to seal this gap between your 3' hydroxyl and 5' phosphate group on the adjacent Okazaki fragment. So reviewing DNA replication, there's initiation, elongation, and termination. So what we've covered so far is the initiation and elongation, both the lagging and leading strand. Okay, reagents that you need for this, you need the DNA polymerases, you need DNA ligase. DNA polymerase III would be the leading strand, DNA polymerase I would be the lagging strand. Ligases to seal the lagging strand. You need helicases to unwind the DNA, topoisomerases to alleviate the strain, and you do need nucleotide triphosphates and dinucleotide triphosphates. And then the primase is what creates the primers for the lagging strand. We've talked about that nick translation, just the fact that a ligase will seal the nicks on the Okazaki fragments. And then we've talked about fidelity, and specifically that coming mainly, well, first from the polymerase active site, and then from the exonuclease activity that polymerase has. And table 25.1 in the book shows the 3 different DNA polymerases from E. coli. Their genes are shown there. Number of subunits. Do they have 3' to 5' exonuclease activity? Yes, they all do. 5' to 3' exonuclease, only one of those has that, and that's DNA polymerase I. And then the polymerization rate and the processivity. How are the rate and processivity measured? So you can visualize this using single molecule DNA replication with a fluorescence microscope. And so that was done by this group. And if you want to look at that, the links there. and it, not obviously not going to be on an exam or anything, but it's just kind of neat to see this. So how does replication stop? Well, that's what's referred to as termination. And this occurs when the helicase gets to a specific DNA sequence on the chromosome called a termination site, or TER, and that's shown here in this E. coli chromosome. So it's either where you meet this termination site, or you can think of it as well as if your two replication forks meet, that's obviously going to stop your replication. So when that happens, you're going to be left with two chromosomes that are wound together. So if we're thinking about bacterial chromosomes being replicated, you're going to have two circles of DNA, and they're going to be tied together. So that's where another spot where your topoisomerase is used to unwind and detach these two from each other. Okay, and when this happens, your chromosome has been fully replicated. And this is just our example of what happens when you reach your end of replication. When the two forks meet, right, your two newly synthesized chromosomes are interwound together, and that's where you need that topoisomerase to separate them. Okay. So far, we've only kind of looked at prokaryotic replication. Eukaryotes are similar, but obviously more complex. They have many polymerases, not just three. They have a leading and lagging strand, so that's similar. They also have helicases and topoisomerases, which are similar. They have origin of replications, but there are many of them, and they have variable sequences to them. The reason why they have so many is because their genomes are quite long, and the chromosomes are much, much longer than bacterial chromosomes. So if you only had one origin of replication on each chromosome, it would take too long to replicate them. So you need many origins of replication. Right? The DNA is linear, not circular, so that also will make the replication process slightly different. And specifically, the termination is going to be different. And that's also one of the reasons why telomeres are important, because they're on both ends of the chromosome. Eukaryotes, it's estimated the number of nucleotides you can do when you replicate this is 50 nucleotides per second. Okay, so if you didn't have more than one origin of replication, you could see how that could take quite a while. And this is in a comparison to the bacterial polymerase and replication, sort of shown here in this figure that we've kind of looked at already. Right? We have this ORC complex that binds to our origin in the eukaryotic DNA, and then the protein CDC6 comes in, and then our MCM2 will come in, and this is like our helicase protein. Okay. So this complex, the ORC in CDC6, is really used to just load our helicase onto the DNA. And that helicase there is MCM2. All right, so this would be referred to as a pre-replication complex. This is just from another book, a different way to look at that. Okay. Then our helicase can go both directions, right? We have our polymerase alpha, which is polymerizing the strands, and you can see how this gets much more complex. There's more proteins involved. I think this is a good figure to actually show this, because it looks a little bit like the figure for prokaryotic replication that we've been looking at. So MCM, again, remember that is the same as helicase. It's what's unwinding this. The leading strand, we have polymerase epsilon. The lagging strand, we have polymerase delta. These purple squares are just our histones. Remember, histones, our DNA in eukaryotic cells are wound around histones, so those have to be dealt with as well. That's one of the reasons this becomes more complicated. Okay. If we look at some of the polymerases in eukaryotic cells, they're shown here. Alpha and beta are both nuclear polymerases. Gamma's mitochondrial. The delta and epsilon are also in the nucleus. And if you look through this in a little bit more detail, the alpha is the one that replication initiation. It's responsible for that. The beta is for DNA repair specifically. It's really the delta and the epsilon that are important, or that are similar to the polymerase that we've seen in E. coli, right? And so delta is lagging strand and epsilon is the leading strand. And they both as well have that 3' exonuclease proofreading ability. And a summary table here that shows you the prokaryotic proteins and compares them to what the similar proteins are in the eukaryotic system are shown here. Okay, so for this class, knowing prokaryotic replication is important in being able to, explain that in some detail. For eukaryotic replication, just being able to know which proteins are similar, I think, is important, and specifically the two polymerases and the helicase protein. Okay, so that kind of clears up the first section of chapter 25. Moving on to the second section, DNA repair. Damage to things like RNA and protein are not so important because RNA and protein are constantly being degraded and resynthesized within a cell. So if you get something that damages them, no big deal, because it can just be degraded and replaced. But DNA is not replaceable. DNA stays there essentially forever, unless you're replicating it for a new cell. So when damage does occur in DNA, it needs to be fixed. And it needs to be fixed before it's replicated. So if there's damage there and you have a different, let's say a different base pair comes in, a mutation, if that's not repaired before replication, that mutation will be passed on to the next daughter cell line. And because of that, the cells spend a lot of energy in the form of ATP to correct these mutations. So at a molecular level, damage usually involves changes in the structure of one of the strands. Okay, the vast majority are corrected by a repair system using the other strand as a template. Okay, so you have a mistake on one strand, you can always use the other strand to repair that as a template. Okay, so some base changes do escape the repair, and then their incorrect base serves as a template in replication. Right, and when that happens, the daughter DNA carries that change sequence in both strands, and that's what we refer to as a mutation. accumulation of mutations in eukaryotic cells, it's strongly correlated with cancer. And most carcinogens are also mutagens. So most chemicals that cause cancer, they're doing so because they are causing mutations in the DNA. And again, cells expend a lot of energy for repair. So let's look at different kinds of damage. Types of damage. You can have a single nucleotide mismatch, right? That may change an amino acid. Sometimes they don't, but oftentimes they do change the amino acid. So that in that sense, you have a substitution of one base for another. So those are typically termed as a missense mutation. You can have an insertion or deletion of an entire gene or a piece of a gene. You can have a frame shift, which removes or inserts a small amount of a gene somewhere. And then you can have mutations also in the intronic regions. And those can affect the splicing of the gene, so how it's cut, how the exons are cut and put together. So those can be quite damaging. If a mutation has no bad side effects to it, it's known as a silent mutation. Those can also be referred to as polymorphisms. Sometimes the mutations do give an advantage, right? Because this is sort of a random process going on. So sometimes you can have a mutation that makes the protein better in a certain environment. And that's kind of the basis of evolution. Okay, that's a figure from the book showing how a bacteria strain can evolve in a environment to a mutagenic environment. All right, so let's look at the four types of DNA repair systems. The first one we'll talk about is covering mismatches. Okay, and these arise from occasional incorporation of incorrect nucleotides. So you have the wrong nucleotide being incorporated in, that's a mismatch. You do mismatch repair for that. Okay, the second one is for abnormal bases. Those arise from spontaneous deamination reactions or certain chemical reactions that occur. And to repair those, you have to actually cut those out and insert back in the correct base. Backbone lesions, those occur from exposing to radiation. And again, you have to cut those pieces out to repair them. And finally, direct repair is needed to reverse photo damage or alkylated DNA. A lot of DNA in bacteria, anyways, is methylated, the parental DNA. So DNA is methylated before replication at specific sites, and those are with the sequence GATC. The newly synthesized strand is unmethylated for a short period after it's synthesized. Right? And the newly synthesized strand becomes methylated before the cells divide. So this methylation is thought as a way to ensure that cells do not divide before replication is complete. And so that's one of the ways that this mismatch repair, it utilizes this fact. So the methylated strand is thought of as the parent strand or the template strand. So that's the strand that's going to be used for mismatch repair. Okay, in this case, we have our parent strand here. It's methylated at that GATC site. Right? And our newly synthesized daughter strand, you can see here. And at this highlighted blue spot, we have in the daughter strand, we have a mismatch that has been incorporated into it. Right? The template strand has a C, but in our newly synthesized strand, we put an A. So that doesn't match up. So for mismatch repair to work, what's going to happen is it's going to use the methylated strand as the template and repair that mismatch base pair. Right, and this just shows how when you get replication, the parent strands are replicated. For that short period of time, the daughter strands are not methylated, and that's just referred to as hemimethylated DNA. Okay, so our repair machinery recognizes the shape changes. So a mismatch base pair, it's not going to hydrogen bond the same, and because of that, there's going to be the shape is going to be slightly different. So your machinery to repair this is going to look for that change in shape. And specifically, these are the mut-L and mut-S complex. Okay, it finds that mismatched shape. It binds to there using ATP. Okay, and when it binds there, what happens? It actually cleaves the unmethylated strand, so the daughter strand is cleaved. It kind of makes this bubble here with that mismatch in the center of it. It cleaves that daughter strand, and then it will degrade the unmethylated DNA strand to the point of the mismatch. The cleaved strand with the incorrect nucleotide is degraded by exonuclease, so one at a time, and then it's rebuilt by a DNA polymerase. This, at least the time these slides were made, it's unknown how the correct strand of DNA is identified in the eukaryotes because you don't have the GATC methylation sites in eukaryotes. Those are only in bacteria. So it's really sort of unclear how eukaryotes go through this process of mismatch repair. Okay. Right, so we can see here this mut-L, mut-S, mut-L, mut-H complex. We'll cut that open, excise out that DNA, and then polymerases fill it in. Method 2 is base excision repair. So actually cutting out a damaged base. So when a base has been damaged either from losing an amine group or other small structural changes, it needs to be cut out of the DNA and replaced with the proper base. And the enzyme used for this is DNA glycosylase, and that cleaves that N-glycosidic linkage. So our DNA bases, we have a sugar, and from that anomeric carbon, that sugar is bonding to a nitrogen group on our base, so that's what's referred to as an N-glycosidic linkage. So that bond between our ribose and our base is cleaved. And when that happens, it leaves what's known as an AP site, or apernuric site, which simply means you're missing a purine or missing a pyrimidine site. So AP just means you're missing the base. Right, so if we look here, our damaged base is shown in this figure right here. Okay, that damaged base, our DNA glycosylase will identify, and then it removes just the base. So the sugar, the ribose sugar, and that phosphodiester linkage in the chain is left intact. All right, so we just are missing the base, and so that's what our AP site is. Okay, no base there. Okay, then our AP endonuclease comes in and sees that AP site, and it then cleaves that phosphodiester bond at that point, Some specific examples are shown here. So the backbone is actually removed there, and DNA polymerase 1 will then come in and add new nucleotides. Right, so this is fairly similar to Nick translation, where DNA polymerase 1 is removing RNA primers and inserting a new small sequence of DNA there. The same process, it's going through the same process. And in eukaryotes, it's done by a special polymerase that's doing this. And then at the end, you're going to need a DNA ligase because there'll be a nick there. Third method is nucleotide excision repair, and this is where you have, it's sort of similar to the previous method, except now we have something that it's not just one base that's altered, but it's several bases in a row that's altered, something that's referred to as a lesion, right? And so lesions kind of create larger distortions of structure. So in E. coli, we have what's known as an exenuclease. It will find this lesion and cut out a larger piece of it around there. And in this case, it's 13 nucleotides long. Okay, so the exenuclease cuts those, helicase will unwind it, DNA polymerase 1 fills it in, and then your ligase terminates that and seals the nick. In humans, it's a similar process. You have what's known as exenuclease again. It's cutting out 29 bases instead of 13. Helicase again unwinds it. DNA polymerase epsilon is the one that's filling it in, and you get a DNA ligase to seal that nick. The last category of these direct repair mechanisms, things like photolyases, methyltransferases, or this ALK-B transferase. We're not going to really talk about any of these specifically. Some of the examples are shown here on the slides and in the book, but you won't be responsible for this in this class. Okay, so there's a direct repair mechanism for methyltransferases. Photolyases use flavin cofactors to help facilitate this. Okay, the last repair mechanism we'll talk about is what's known as the SOS response pathway. This is when there's damage at a replication fork that causes it to sort of stall. And how the cells get around this is they use a more error-prone way to synthesize the DNA just to kind of break through this. And that's because, the introducing errors is bad, but if the replication fork stalls and it can't replicate, that's even worse, okay? So having, half a chromosome being synthesized is much worse than having one that has more errors in it. So they use these proteins, these polymerases that introduce more errors. And in bacteria, it's about an error every 1,000 nucleotides. So that's something like 100 times less fidelity. Okay? In eukaryotes, it's the same way. You won't be responsible to know any of these exact polymerases, but just understand that the SOS response pathway is a way that replication can still occur even though there's some major damage. And that moves us into the third section, DNA recombination. And this is another way in which DNA can be repaired, as we'll see. Segments of DNA can rearrange their location within a chromosome or from one chromosome to another. And three mechanisms of that are homologous recombination, site-specific recombination, and DNA transposition. We'll see examples of each of these. Don't really fret about the exact details of these, because some of them, as you'll see, can seem pretty complicated, but just try to understand the differences between the three. And these processes occur with one respect to repairing of DNA. So these are ways that some pretty big damage in DNA can be repaired. It helps in the segregation of chromosomes during meiosis, and it is also a way, it's thought of as a way to enhance genetic diversity. So the first one, homologous recombination. This is a genetic exchange occurring between two molecules that have an extended region of very identical sequences. So things like pairs of chromosomes in diploid cells would have large regions of nearly identical sequences that this recombination can happen. Okay. Eukaryotes, this occurs in meiosis. In E. coli, this process is used primarily for repair. So if we look at our two chromosomes, a homologous pair of chromosomes, they have, again, they're almost identical, share very large portions that are almost identical in sequence. So you can think of it as they can kind of connect together and then switch which part is where. So if you look at here, you have the light blue and dark blue in your homologous pair. If those kind of connected together and then swapped, now we have these light blue ends on the dark blue and dark blue ends on the light blue. Okay. And again, this is used to repair DNA damage and also thought of as a way to enhance genetic diversity. Looking more specifically at how this occurs, and it's through what's known as holiday junctions. Right. Don't need to know many of the details of this. I'm not going to be asking questions for you to give me the details of this, but know that holiday junctions occur in this homologous recombination. Okay. And specifically what's happening is on one strand you can get a cut, and then an exonuclease can kind of remove some of those base pairs, and that gives you these overhanging ends, which then can be recombined with the homologous DNA sequences in the other chromosome. Okay, and when you do that, then a polymerase adds that DNA back in. So you're kind of going through DNA replication again, very similar process to fill in those gaps, and you have these crossover points or holiday junctions. And this just shows how these are made in a little bit more detail, right? Helicase unwinds this, nuclease degrades one of the strands, and then you have this overhanging region. Right. And if you have an overhanging region like that, if it's single-stranded DNA, it's pretty flimsy and flexible. One way to assist in this strand, what's called strand invasion, where this single strand can invade the homologous chromosome, is this protein called REC-A. So this REC-A protein, you get multiple copies of this sticking to the single-stranded DNA, sort of like single-stranded binding protein in prokaryotic replication, and it goes on there, and what it's doing is it's making sort of like a filament, or you can think of it as a needle. So it just gives it some stiffer structure that can actually be inserted into the other chromosome, the other double-stranded DNA. Here's a crystal structure of that. This is an electron micrograph of that. So DNA by itself, shown here, it's very bendy and flexible. When it's bound to REC-A, it's much straighter and has more tensile strength, more structure. Okay. Right, these holiday junctions can be cleaved a number of different ways. Not going to need to know exactly how they can be cleaved in this class, but depending on how they are cleaved, you can get different combinations. Genes here are still present in the same order, but genes from one parent can be mixed with the genes from another parent. So this, as you can see, is a way that more genetic diversity can occur. Okay, the second method of this is recombination, is site-specific recombination. And this is limited to specific sequences that are 20 to 200 base pairs long. Okay, important aspect of this method is that there are specific proteins that catalyze this called recombinases, and they use tyrosine in their active site to do this. So what they're doing is the recombinase binds, and it creates sort of like a twist in that double-stranded DNA, and then the tyrosine can cleave that DNA in those twists, and then it reattaches it in a different order. So here we have the blue and the red complementary strands together, and these recombinase proteins are coordinating that. When they cut and then bind again, now you have the blue attached to the red. And so some more detail in how this is occurring is shown here. Not going to need to know the exact mechanism for this class, though. It's similar, these recombinases are similar to the mechanism of topoisomerases, and if you remember, those also had tyrosine in their active sites. Here's an electron micrograph of that, okay, and you get that the same sort of holiday intermediate using this method as well. All right, and the final method of recombination is DNA transposition, and this is where the transposons come into effect that we mentioned last lecture. Right, these are very mobile pieces of DNA. And they carry itself, or they can also carry some copies of genes. Right? They have on the outsides of them some terminal repeats, and they do not need to interact with homologous sequences. So these can kind of move around more freely than the last two cases that we're looking at more homologous type sequences. Right. This is the Book's figure to show how transposons can move around and be inserted into new places in the gene, or excuse me, new places in the chromosome. Okay. Again, the uses of recombination, repair, that's specifically in DNA repair where the error includes double-stranded breaks or double-stranded cross-links. So those are some pretty big damage to DNA, not just a simple mismatch, but where you have something that would stall replication. Okay. Places where both strands are missing or both strands are altered, okay, again, major damage. And then certain lesions that are pretty damaging. And the BRCA2 gene is actually involved in this type of repair. And then genetic diversity, that's another use of recombination. And specific example of that the book talks about is the IgG, which is the immunoglobulin, a protein that's involved in antibody production. Right, so how does recombination help in repairing DNA? Well, if you have a very big lesion in your DNA, what can occur when you're trying to replicate that, once you get to that lesion, your polymerase can stall, okay? And it stalls, and then you have to go through the more error-prone sort of repair mechanism for that. If you also have something like a double-stranded break that's not repaired and you're trying to replicate this, again, you're going to get a polymerase. When it comes to that, it's going to stall. And to do that, you would need, in both of these cases, the the recombination repair can help fix these. So if we look at lesions specifically, right, when the replication fork gets there, what can happen is that you get a stall, but if it on the lagging end, the opposite end, it can go through and polymerize some of that, and then it can back out to where that stall occurred. And the daughter strands that have been synthesized can then, you come to a point where they match up, and from there you can start polymerizing again, right? So in a sense, what you're doing is you're using the newly synthesized strand that matched up to this region where your lesion was, and you're using that now as your template instead of this blue strand as the template. Okay, and then you can then go forward again, and now that newly synthesized dark red, or I should say light red, goes over that lesion. So you've bypassed it. Breaks sort of work in a similar fashion, except now we're using the strand invasion with Rec A. So you can use strand invasion from your opposite side to invade that point, that double-stranded break, and use that to continue your synthesis through that. So that repairs the stall. And doing so, you get that crossover holiday junction, which then can be excised out. Those specific enzymes needed for recombination, shown here. The one that I really want you to focus on the most is Rec A. That's the one that binds to single-stranded DNA and allows it to be inserted into double-stranded DNA. Okay, that by far I think is the one to really remember. Right. And then obviously you need helicase and nuclease in one enzyme. Okay, and that's the same as it would be in DNA replication. So BRCA, it's probably a gene that you've heard of before. They're tumor suppressor genes. They encode for proteins that signal or help DNA recombination mechanisms that are used to repair DNA. So they're involved in this recombination process. Okay, if you have a mutated version of BRCA, one of the BRCA genes, your DNA is not always repaired properly. And so in people that have these mutations, cancer can occur more readily. And so especially with like breast cancer, it's a very well-known association between that. And you can actually get screened genetically for this, if you have especially if you have a history of certain cancers in your family. Okay. And some other proteins like BRCA1 and 2, RAD51 is another one of these genes, and this stimulates strand invasion. And then this larger complex here localizes RAD51 to the double-stranded break. So these are genes that you might have heard of before, and they're involved in this recombination repair mechanism. Immunoglobulin is another one that's involved in recombination, and it's involved in a different way. It's using recombination to create genetic diversity, and the genetic diversity that it's important because it allows it to make very many different types of antibodies that can then attack many different foreign invaders into our body. So you use this recombination because you can have many different protein products from the same gene. Okay, so there's about 30,000 genes in the genome, human genome, but you can produce over 1 million different antibodies are encoded. Okay, so these really add a lot of diversity. This recombination events. And if you're interested in the specifics, they're shown here, but in this class, you're not going to need to know how these specifics work. Just that recombination is what's allowing these immunoglobulins to have many different products from the same gene. Right, so in summary from chapter 25, what's important? Replication, the most important. Okay, DNA replication catalyzed by DNA polymerases that use one of the strand as a template while adding new nucleotides to the 3' end of the newly synthesized chain. Okay, we talked about several repair mechanisms that exist to correct mismatches in DNA mutations. So there are 4 main categories. Really, the first three are the ones that we got more specific about. And then finally, recombination, and that's where regions of DNA are swapped either for repair or for genetic diversity.